Life begins when a fertilized egg divides to form new cells, which in turn also divide. Initially, these cells look identical, but over time they begin to change. In a process regulated by our genes, some go on to form the heart, and others, for example, the brain. What are the mechanisms that enable a single cell to become a complex organism? This is one of biology's great questions, and one which the research of Christiane Nüsslein-Volhard has helped to resolve. Her work has allowed us to identify the genes that regulate the development of living organisms, shedding light on broad areas of biology and medicine. In the 1970s, she began to study the developmental genetics of mutant fruit fly embryos and was able to identify the genes that regulate their complete development. Later, she focused her research on the key developmental regulators in vertebrates using a new model organism, the zebrafish. Her discoveries opened different lines of research, some of them in the field of medicine, which, for example, have led to a better understanding of the causes of anomalies in human embryonic
Hi, Yanni. Many congratulations on being awarded your honoris causa in Barcelona. I hope you have a fantastic day, and I very much look forward to seeing you again once this whole pandemic is over. Bye.
Yanni, congratulations on this wonderful award. I just wish you could be there in person. And thank you again for all the wonderful science you've done and wonderful.
Questions, and one which the research of Christiane Nüsslein-Volhard has helped to resolve. Her work has allowed us to identify the genes that regulate the development of living organisms, shedding light on broad areas of biology and medicine. In the 1970s, she began to study the developmental genetics of mutant fruit fly embryos and was able to identify the genes that regulate their complete development. Later, she focused her research on the key developmental regulators in vertebrates using a new model organism, the zebrafish. Her discoveries opened different lines of research, some of them in the field of medicine, which, for example, have led to a better understanding of the causes of anomalies in human embryonic development. Life begins when a fertilized egg divides to form new cells, which in turn also divide. Initially, these cells look identical, but over time they begin to change. In a process regulated by our genes, some go on to form the heart, and others, for example, the brain. What are the mechanisms that enable a single cell to become a complex organism? This is one of biology's great questions, and one which the research of Christiane Nüsslein-Volhard has helped to resolve. Her work has allowed us to identify the genes that regulate the development of living organisms, shedding light on broad areas of biology and medicine. In the 1970s, she began to study the developmental genetics of mutant fruit fly embryos and was able to identify the genes that regulate their complete development. Later, she focused her research on the key developmental regulators in vertebrates using a new model organism, the zebrafish. Her discoveries opened different lines of research, some of them in the field of medicine, which, for example, have led to a better understanding of the causes of anomalies in human embryonic development. Life begins when a fertilized egg divides to form new cells, which in turn also divide.
assistents que es posin dempeus. Gràcies. Senyores i senyors, la cerimònia és a punt de començar. Si us plau, ocupin els seus seients i desconnectin els telèfons mòbils.
Bon dia a tothom. Good morning, everyone. Benvinguts a la cerimònia acadèmica d'investidura com a doctora honoris causa de Christiane Nüsslein-Volhard. Welcome to the academic investiture ceremony of Christiane Nüsslein-Volhard as doctor honoris causa of Pompeu Fabra University. En aquest acte es combinarà el català i l'anglès. Iniciem l'acte amb la lectura de l'acord del Consell de Govern de concessió de títol de doctora honoris causa a Christiane Nüsslein-Volhard. Té la paraula el secretari general Pere Torra. We begin the ceremony with reading of the agreement of the Board of Governors of UPF to grant the degree of doctor honoris causa to Christiane Nüsslein-Volhard. I now give the floor to the Secretary General, Mr. Pere Torra.
Gràcies, rector. Bon dia. En la sessió del Consell de Govern de 19 de febrer del 2020 es va prendre l'acord següent. El Consell de Govern, a proposta del Consell del Departament de Ciències Experimentals i de la Salut, actual Departament de Medicina i Ciències de la Vida, i de la Facultat de Ciències de la Salut i de la Vida, acorda concedir el títol de doctora honoris causa per la Universitat Pompeu Fabra a la doctora Christiane Nüsslein-Volhard, en reconeixement a la seva trajectòria investigadora excepcional que ha permès identificar els gens que regulen el desenvolupament dels organismes vius i descobrir principis generals del desenvolupament embrionari. La doctora Christiane Nüsslein-Volhard és, a més, un exemple d'honestedat i compromís de la recerca científica amb la societat. The UPF Board of Governors, following a proposal by the Council of the Department and the Faculty of Health and Life Sciences, approved to award the degree of doctora honoris causa of the UPF to Professor Christiane Nüsslein-Volhard. The university recognizes the achievements of an outstanding researcher who has contributed to our basic understanding of embryonic development. Doctora Christiane Nüsslein-Volhard is also an example of honesty and commitment of scientific research to society.
Moltes gràcies, senyor Torra. A continuació demano al padrí, el doctor David Comas, que vagi a buscar la doctoranda i l'acompanyi fins al lloc que ocuparà a l'auditori. Thank you very much, Mr. Torra. Now I ask the academic sponsor, Dr. David Comas, to go and find the doctoral candidate and to place her on the stage.
Demanem als assistents que es posin en peu. Gràcies.
Professora Nüsslein-Volhard, sigueu benvinguda a la nostra universitat. Welcome to our university, Professor Nüsslein-Volhard.
Life begins when a fertilized egg divides to form new cells, which in turn also divide. Initially, these cells look identical, but over time they begin to change. In a process regulated by our genes, some go on to form the heart, and others, for example, the brain. What are the mechanisms that enable a single cell to become a complex organism? This is one of biology's great questions, and one which the research of Christiane Nüsslein-Volhard has helped to resolve. Her work has allowed us to identify the genes that regulate the development of living organisms, shedding light on broad areas of biology and medicine. In the 1970s, she began to study the developmental genetics of mutant fruit fly embryos and was able to identify the genes that regulate their complete development. Later, she focused her research on the key developmental regulators in vertebrates using a new model organism, the zebrafish. Her discoveries opened different lines of research, some of them in the field of medicine, which, for example, have led to a better understanding of the causes of anomalies in human embryonic development.
A continuació, la doctora Cristina Pujades, catedràtica del Departament de Medicina i Ciències de la Vida de la UPF, llegirà l'elogi de la doctoranda. Dr. Christina Pujades, full professor of the Department of Medicine and Life Sciences at UPF, will now read Christiane Nüsslein-Volhard Laudatio.
Can we hear? Yeah. And yet, 1 year later, here we are. It is a real pleasure and a great honor for me to introduce Professor Christiane Nüsslein-Volhard in the context of her nomination as doctora honoris causa by the University Pompeu Fabra. Professor Christiane Nüsslein-Volhard does not need much of an introduction. She is one of the best-known scientists in developmental biology and genetics and biology in general. She pioneered a scientific approach with molecular studies in the fruit fly Drosophila, which constituted a revolution in biology and was recognized with a Nobel Prize. To convey the importance of Professor Nüsslein-Volhard's achievements during her professional career is a difficult task, and I decided to do so by giving you my vision of her pillar contributions to this revolution in biology, specifically to developmental biology and genetics, and to comment on her broader contributions to the scientific community. Before entering into the scientific accomplishments, I will briefly summarize the field of developmental biology, which basically aims at understanding how a whole, fully differentiated organism is generated from a single multipotent cell, the zygote. Embryonic development involves an exquisite balance between cell proliferation and cell differentiation that results in an enormous number of different cell types that must occur in their correct proportions and locations. The cartography and final shape of the organism are established by sequential processes that convert initially very simple patterns in others of increasing complexity. Thus, the building of a new organism involves the precise coordination of growth, spatial patterning, and morphogenesis. The result is the generation of an individual with many different specialized organs, each with specific shape and function. You may take this as a brilliant triumph of evolution. Understanding the mechanisms that control the embryonic development in different animal species and the roadmap that allows this to be highly reproducible in every individual must be based on solid mechanisms selected throughout evolution. Getting deep knowledge about this mechanism is a crucial key for tissue biology, regenerative medicine, and biomedical engineering that aim at the therapeutic reconstruction of tissues and organs. These fundamental questions about how an embryo develops into a new individual were posed long ago by the ancient Greek scientists and philosophers. In the last century, advances in experimental embryology, the tradition of watching and manipulating embryos that had started in the late 19th century, joined the application of genetics and gene manipulation. With the birth of molecular biology and its development into a fully developed discipline, these fundamental questions became addressable. Much of the excitement in developmental biology nowadays comes from our growing understanding of how genes direct these developmental processes, and it was at this crucial crossroad, envisaging the importance of placing genetics at the central stage of embryonic development, where Professor Nüsslein-Volhard played such a crucial role. Professor Nüsslein-Volhard has been interested for many years in how complexity arises from basic patterns, and she did not fear in undertaking immense challenges to pursue this question. She began by studying gene transcription, how genes are switched on. During her PhD in the University of Tübingen, she worked in the structure of gene promoters and isolated RNase polymerase binding sites from a phage, a bacterial virus. At that time, techniques were still developing and were far less powerful or as fast as today. However, Professor Nüsslein-Volhard saw the potential of molecular biology for understanding the complex mechanism underlying embryonic development, one of the pillars of her scientific contributions. This was how, after her PhD, she transitioned to developmental genetics. In those years, it was recognized that the steps producing morphological complexity in different organisms were reproducible from one embryo to the next, and that genes and cell interactions were important in embryonic development. But the underlying mechanisms were largely unknown and somehow, if not, enigmatic. Professor Nüsslein-Volhard predicted that developmental mechanisms were so basic that they had to be evolutionarily conserved, and that genetics would lead the path to the logics of the biological systems. This is, in my opinion, one of the most revolutionary ideas in biology and another of the scientific pillars that pave the future road to travel. Professor Nüsslein-Volhard wanted to go beyond the evident and thus investigated the logics of constructing the embryo. For this purpose, she aimed at combining genetics and developmental biology. The rationale was that the function of a gene can be deduced from the effects produced by its deletion, a simple but powerful concept. Genes whose mutations result in similar phenotypes are predicted to have similar functions, and their products are likely to cooperate. And brilliantly, she foresaw that knowing how simple organisms form would help us to better understand how complex organisms develop. This is how, in 1973, she moved for a postdoctoral stay to the laboratory of Walter Gehring at Biozentrum in Basel, who had just published the first set of early mutants in Drosophila melanogaster. No doubt that the Biozentrum and the Gehring's lab was one of the places to be if you wanted to understand genes and development. Developmental biology was flourishing in new concepts such as tissue pattern, morphogen gradients, positional information, and Drosophila was a fantastic and rather simple and non-expensive model system to work with. As she stated in her Nobel Prize biography, it was difficult to be a beginner in everything after having been an expert in almost everything in the previous lab. With a whole new set of techniques being established and surrounded with talented colleagues, she studied the bicodal gene, which is today one of the best-known genes. While there, she developed new methods to generate her own mutants. It was then that she met Eric Wischhaus, who just had finished his PhD, with whom she would share the Nobel Prize, together with Louise years later. In 1978, Professor Nüsslein-Volhard and Professor Wischhaus got a job offer from the Director General of the European Molecular Biology Laboratory, the EMBL, to establish a joint lab. Together, they started a new project, conceptually very simple, extremely powerful, and technically not so obvious. The idea was to identify all genes involved in embryonic development by mutating them at large scale. In other words, to mutate all the genes of the genome in such a manner that not more than a single gene would be mutated in each individual, and then to look for specific developmental defects and trace back their function, what we now call reverse genetics. That was the first large-scale mutagenesis genetic screen in Drosophila embryos, the Heidelberg screen, performed between 1979 and 1980 at the MBL in Heidelberg. Let me give you a flavor of it. Since they wanted to understand the logics behind cell determination and cell differentiation, as well as to trace back the decisions made by cells and tissues during early development, it was necessary to find out how many genes were involved in embryonic pattern formation, whether each of the genes was unique, and what types of changes and alterations can be traced back to a mutation in a single gene. Large-scale mutagenesis experiments would allow the identification of most, if not all, genes affecting embryonic developmental decisions on the basis of visible mutant phenotypes in the larva. With this in mind, they generated a collection of 600 Drosophila embryonic mutants that corresponded to 120 genes. They shared this mutant collection with the scientific community, pioneering the exchange of materials and data among the fly labs. That work was published in the journal Nature in October 1980 and set up a turning point in the field, beautifully demonstrating that genes control the logics of embryonic development and that there was a sequential gene hierarchy. Those mutants subsequently provided a very rich resource for understanding many fundamental processes in development and disease. Most of the Heidelberg genes are now molecularly characterized, and many of them are shown to be conserved in other animal species, including humans. Although the screens were initially driven entirely by curiosity, the mutants now serve as model for many human diseases, showing once more the importance of fundamental scientific research for health sciences. After a few years, Professor Nüsslein-Volhard was appointed to the Friedrich Mischer Laboratory of the Max Planck Society in Tübingen, where she moved in 1981, while Professor Wischhaus moved to Princeton University. But they maintained their ongoing scientific collaboration, which resulted in exciting discoveries and impressive breakthroughs, such as the discovery of the genes involved in the establishment of the fly-body axis or the role of morphogens in development. These investigations gradually evolved to a far more comprehensive understanding of the principles of axis determination in animals. Overall, this innovative work led to the award in 1995 of the Nobel Prize in Physiology and Medicine to Professor Nüsslein-Volhard and Professor Wischhaus for the discoveries concerning the genetic control of early embryonic development, together with Professor Luis, and revealed that embryonic development was actually controlled by a relatively small fraction of genes within the genome, the so-called developmental genes. Although impressive, this is only half of the story. One big question emerged after the fly work. Were those genes and mechanisms extensive to other animal species? In particular, do vertebrates use similar strategies? Around that time, high degree of genomology among the metazoans slowly became apparent. As Stephen Jay Gould stated in 1977, I also believe that an understanding of the regulation must lie at the center of any rapprochement between molecular and evolutionary biology. For a synthesis of these two biologies will surely take place, if it occurs at all, on the common ground of development. Thus, in the middle of her success in the discovery of the molecular mechanism governing Drosophila embryonic development, Professor Nüsslein-Volhard went a step further and moved out of what now we may call the comfort zone. She explored whether a fish, such as a zebrafish, could eventually be exploited like the fly for the genetic analysis of vertebrate developmental genes, and decided to test whether it could be used for the same strategy as for Drosophila, a large-scale genetic screen. In doing so, she anticipated zebrafish as one powerful vertebrate model organism for genetics. For those not familiar with what a wet lab is, let me tell you that such decision implies an enormous scaling up in your facilities and another level in managing complexity. Surely a big enterprise. but she took it as a fascinating challenge that resulted in the beginning of the zebrafish large-scale screening, or the Tübingen screen, in 1992. There were only 2 other places in the world setting up this model system, in Eugene and Boston. The screening turned out to be much more complex than in Drosophila, as the fly has in total 5 chromosomes, while the fish has 25. Moreover, to reach mutation saturation in practical terms, it was needed to screen at least 3,000 inbred families. This was indeed a titanic project and required commitment and cooperativity to a degree that was exceptional for scientists normally used to work independently on their specific projects. After working during almost 2 entire years, the Tübingen team was able to analyze 1,163 fish mutants that displayed a specific phenotype. This vast enterprise was published in an issue of the journal Development in 1996, which was fully devoted to zebrafish. 22 out of 37 articles came from the Tübingen screen. I still remember attending a seminar at Harvard Medical School in the 90s in which there were 2 speakers, Professor Nüsslein-Volhard and Professor Nancy Hopkins from Boston. They were sharing their new screening adventures with the scientific community. I was fascinated, not only because it was completely uncommon to see 2 women scientists as speakers at that time, but because I had never seen before a seminar with 2 so smart and well-experienced co-speakers talking about those kinds of challenges. Theirs resulted in a very fruitful enterprise, not only for them, but for all of us. Many labs around the world are using today's zebrafish as a model system to study not only vertebrate development, but as patient avatars for cancer biology and precision therapy. And further, they demonstrated that the basic mechanism in the construction of an organism are pretty much conserved among species. Despite our pretenses, we humans are not so singular in this matter, and probably also not in others. As you may anticipate, the impact of this work has been enormous. Now we use simpler vertebrate organisms to study fundamental questions, and from there we can better understand human health and disease. Again, pioneering studies in basic research leading to a dramatic impact in biomedicine. After this brief scientific profile, I would like to comment on the role played by Professor Nüsslein-Volhard as a scientific mentor. As a glimpse, the Nüsslein-Volhard lab style has always been that postdocs leaving the lab took their projects with them, and sometimes PhD students too. Her people may have left Nüsslein-Volhard's lab with some of the most interesting projects that anyone would die to keep for him or herself. This is named generosity, and it is also a bold declaration for the implementation of young scientists as group leaders. For the Tübingen screen, she built a highly committed team of exceptional scientists to work in a communal project for the greater good. She inspired experienced scientists and PhD students to work for the collectivity instead of doing it as independent fellows. In the competitive environment of today's science, this requires to generate confidence among the members of the team and to build up strong bonds between them and the group leader. Everybody needs to trust each other and the director, and this bond is not easy to build. As she stated in an issue of the journal Development in 2012, where she described the Tübingen screen, people had worked for several years on this communal project without any publication, that it had to be ensured that they received proper credit for their efforts. How to deal with the need of publishing the work of so many people and give justice to everybody? How to prevent a race with the labs in Boston, which had similar goals and approaches? These questions are still present today in many labs. Science can be merciless, and if you do not make the right decisions, you risk your work to be forgotten and eventually your career. But coming back to Tübingen, what happened there after the submission of the articles to Development was a wonderful party celebrating the successful end of an immense and unique enterprise. Professor Nüsslein-Volhard has mentored many leading scientists now running their own labs in Europe, United States, or Japan. Those mutant fish provided a rich resource for many laboratories and made of Tübingen a landmark for developmental biologists. We all know that doing science is not such an easy job nowadays. However, her example and advice for young scientists are, in my opinion, extremely valuable. Change fields after your PhD, develop your independent profile, be self-critical, and assess your own abilities in comparison with others. Note that all the above rely on yourself. Finally, I will not end without a brief note on the impact of Professor Nüsslein-Volhard's commitment to closing the gender gap in science. As we all know, or at least we should, women are underrepresented in leading scientific or research positions when measured by their scientific potential. Professor Nüsslein-Volhard, as all pioneering women, worked in an environment where most of her peers were men. She experienced the difficulties of not belonging to the club, but thanks to her determination and intelligence and the many scientific contributions mentioned above, she succeeded. It was witnessing and experiencing the gender inequalities that in 2004 prompted her to initiate a foundation that gives support to talented young women scientists who have children, with the aim to increase the contribution of highly qualified women to edge science in Germany. This foundation, the Christiane Nüsslein-Volhard Stiftung for the Promotion of Science and Research, aims to help to prevent the loss of talent by improving women opportunities in sciences. Her foundation targets specifically the problem of balancing family duties with the strong demand required to develop a career as an independent researcher, and focuses on PhD students and postdoctoral fellows in the fields of experimental natural sciences and medicine. Professor Nüsslein-Volhard knows well that being a scientist is a very demanding professional decision, which very early on requires a high degree of independence, a great deal of engagement and motivation, as well as talent, originality, and intelligence. In order to gain an independent and permanent position, it is necessary strong dedication. This may not be possible if family responsibilities are too big or rely only on women's side. It is therefore not uncommon for a woman to subordinate her career to that of her partner, or to decide against having children. Such a decision is normally not demanded from men. It is usually inconsequential to their career whether they have a family with children or not. Professor Nüsslein-Volhard considered that these were weighty causes for few women in leading positions, and for those who are to more likely remain without children. The objective of the foundation was to provide funds intended to pay for assistance in household tasks and for additional childcare, to relieve these young female scientists from household burden and gain time and flexibility for research activities. She was generous enough to start the foundation with her own funds that seeded for several institutions to join her initiative. I'm happy to say that this foundation has inspired institutions among us, like the Center of Genomic Regulation and our own Department of Medicine and Life Sciences, that pioneered in Spain the establishment of this kind of allowances for young female scientists. This path has been later followed by the Barcelona Institute of Science and Technology and by the rest of the University Pompeu Fabra. These fellowships help, but clearly do not solve things. In other words, there is still a long road to travel, and we all need to keep working with multiple actions and policies to help closing the gender gap. I would like to finish by emphasizing the importance of Professor Nüsslein-Volhard as one of the intellectual pillars of developmental biology, a pioneer scientist who explored and developed new concepts and intellectual frameworks, a leader who promoted long-term projects of great value and mentored top scientists, and above all, an extraordinary researcher who worked for making science a space for intellectual debate where all of us may stand. Thank you. Thank you very much.
Moltes gràcies, doctora Pujades, per aquesta magnífica laudatio. Thank you very much, Dr. Pujades, for this extraordinary laudatio. Demano ara al padrí que acompanya la doctoranda fins a la taula presidencial. I ask the academic sponsor to accompany the doctoral candidate to the presidential table. Pel Consell de Govern de la Universitat Pompeu Fabra heu estat nomenada doctora honoris causa en testimoniatge i reconeixença dels vostres rellevants mèrits. En virtut de l'autoritat que m'ha estat conferida, us faig lliurament d'aquesta medalla, símbol d'aquest títol i del diploma acreditatiu. By the Board of Governors of Pompeu Fabra University, you have been nominated Doctor Honoris Causa in testimony and recognition of your relevant merits. By virtue of the authority that has been conferred on me, I hereby award you with this medal, a symbol of this title and of the supporting diploma. Ara ve el més difícil. Sí. Okay. Here. Come here. We continue with the ceremony. Perquè us heu incorporat a aquesta universitat, rebeu ara, en nom del seu claustre de doctors, l'abraçada de fraternitat, ho farem en format Covid, dels qui s'honoren i es congratulen de ser els vostres germans i companys. As you have been admitted to this university, you will now, in the name of the University Senate, receive an embrace in COVID format of fraternity from those who are honored and pleased to be your brothers, sisters, and colleagues. We can do this, just this. Té la paraula la nova doctora i la convido a fer el discurs d'acceptació. Now I give the floor to the new doctor.
Dear Rector, dear Dean, members of the faculty and students of the Universitat Pompeu Fabra, dear Dr. Poyades, thank you very much for your kind words. And I would also like to thank the musicians and the artists who made this very pretty picture. Ladies and gentlemen, I'm very honored to receiving this doctorate from this young, modern and international University Pompeu Fabra. Thank you all very much for your support. I am a biologist and spent most of my academic career in Tübingen, a small German university town. I've been a director at the Max Planck Institute for Developmental Biology since 1985 and retired in 2014. Most of my research has been devoted to the identification of genes controlling development, initially in the embryo of the fruit fly Drosophila melanogaster and more recently in the zebrafish Danio rerio. Since 2014, I'm running a small research group investigating the development and evolution of color patterns. I will present a summary of my recent research on this topic in a seminar tomorrow. As much of you already have heard from Dr. Puyades, so it will be a little repetitive, but it's the same person, you know. So I cannot invent something new now for you. I studied biology, physics, and chemistry, got a diploma in biochemistry at Tübingen University, and did my graduate work on molecular biology at the Max Planck Institute for Virusforschung in Tübingen. When I was searching for my own research topic, the problem of morphogenesis began to fascinate me. At our institute, the group of Alfred Gira studied Hydra as a developmental system with the aim to isolate factors controlling regeneration in this simple animal. At this time, experimental approaches to study mechanisms of development in higher animals were very difficult and frustrating. At the same time, another group around Friedrich Bonhoeffer studied genetics of DNA replication in E. coli, the bacterium. This successful project convinced me that the genetic approach is very powerful to dissect complex processes. It might also work for the study of development. So I looked for an organism that allowed to study development with the methods of genetics. In the mid-1970s, Drosophila seemed the best choice for applying genetics to problems of developmental biology. It was the model organism in which the basic principles of chromosomal inheritance had been worked out. A small number of mutations had been identified that caused alteration in development. I joined the laboratory of Walter Gehring at the Biozentrum in Basel with a long-term goal to discover morphogens in Drosophila. During my postdoctoral time, 1975 to 1978, I got familiar with Drosophila genetics and embryology and developed methods allowing large-scale screening for mutations affecting embryonic patterning. My first independent position was at the EMBL in Heidelberg, when I shared a lab for 3 years with Eric Wischaus, whom I had met in Basel. Eric and my common interest concerned segmentation of the Drosophila embryo. At the time, virtually nothing was known about segmentation and the determination of segment number in any organism. We set out to systematically screen for mutations affecting segment number and polarity. For the mutant screen, we choose a single dominant tissue of the larval body, the skin, with its cuticle. Mutations that affect patterning could be distinguished from those required for more general housekeeping functions by direct inspection of the larval cuticle patterns of mutant embryos. In our saturation screens, in which we were later joined by Gerd Jürgens, we scored embryos from about 20,000 inbred lines and identified 20 genes affecting segmentation. We also identified a total of 120 genes with essential and important functions in the development of the pattern of the larva, representing 2.5% of all genes mutating to lethality in Drosophila. This mutant collection provided the material for the emergence of a new, rapidly expanding field, developmental genetics, studied in many laboratories worldwide. Its success was speeded up by the development of recombinant DNA technology, which allowed the molecular cloning of the genes we had discovered. It was found that many encode transcription factors, but also the components of most important signaling systems operating in all animals, such as, for example, wingless and hedgehog, were contained in our Heidelberg collection. By and large, the development of the embryo is controlled by a series of transcription factors distributed as molecular pre-patterns. By regulating each other, these patterns become refined until the molecular pattern directly determines the morphological pattern. From the EMBL, I went to the Friedrich Miescher Laboratory of the Max Planck Gesellschaft in Tübingen, and we run an independent junior research group until 1985, when I was appointed director of the MPE for developmental biology. The position, At the FML, we focused on genes determining the informational content of the egg cell operating in the maternal organism that produces the egg. We and others, notably the laboratory of Madame Gans in Gif-sur-Yvette in France, and Eric Wischau's and Trudy Schipper's laboratory in Princeton, discovered about 30 maternally required genes that are involved in the axis determination of the embryo. The anterior-posterior axis is determined independently from the dorsoventral axis, as mutations affect either the one or the other, never both. We discovered bicoid and Oscar, and the group of 10 genes determining the dorsal-ventral axis, including the genes dorsal and toll. In transplantation experiments, we found that the localized RNA products of bicoid and Oscar were required to form 2 organizing centers with long-range activity localized at the anterior and posterior of the egg, respectively. Bicoid encodes a transcription factor that is produced at the anterior tip of the egg from its pre-localized mRNA and spreads posteriorly to form a concentration gradient. Artificially changing the gradient results in a shift of the pattern. This was the first demonstration of a morphogen, a protein determining the pattern in a concentration-dependent manner. We also unraveled the signaling systems determining the dorsoventral axis with 11 components, including toll as membrane-bound receptor and Spätzle as extracellular ligand. A morphogenetic gradient of dorsal determines different regions along the dorsoventral axis. However, the gradient is formed in this case by differential uptake of the dorsal protein into the blastoderm nuclei. Many excellent scientists have contributed to these findings, and I would like to mention Ruth Lehmann, Hans-Georg Frohnhofer, Wolfgang Driever, and Daniel St. Johnston, who worked on the patterning along the anterior-posterior axis, and Catherine Anderson, Dave Stein, and Siegfried Roth for their contribution to the understanding of the dorsoventral axis. Drosophila, as an insect model organism, has rather special properties. It is in many respects very different from vertebrate animals. Therefore, it was not clear a priori to what extent the results obtained in Drosophila could be generalized and how much we could learn from them for an understanding of the development of vertebrates. When George Treisinger chose the zebrafish, a small freshwater cyprinid, as a vertebrate model organism to study development, I got fascinated by this animal. Zebrafish has transparent embryos which are rapidly developing outside the maternal organism and that thus ideally suited for in vivo inspection of embryonic development. We performed, this time as a collaborative project of 12 scientists, a large-scale mutagenesis experiment in which we isolated 1,200 mutants affecting the morphology of the embryo or larva at successive stages of development and defined over 300 novel genes. Affecting many aspects of early development, such as gastrulation, segmentation, muscle formation, development of brain, heart, liver, skin, fin, and sensory organs, which were followed up by collaborators establishing their own labs elsewhere. Work in my laboratory focused on traits that are distinct between insects and vertebrates. We have been investigating processes of cell and tissue migration in the lateral line nerve as well as in microglia, neural crest development, the development of the skin and structures shaping the adult body, such as fins and scales. Our present project in my small emerita group focused on the development of the striped pigment pattern of the adult zebrafish, which has established itself as the prime model for color pattern formation in vertebrates. Color patterns are prominent features of most animals. They are highly variable and evolve rapidly, leading to large diversities between species, even within a single genus. They are targets of natural as well as sexual selection, but despite their high evolutionary significance, little is known about their development and evolution. The zebrafish, Danurerio, displays a conspicuous pattern of alternating blue and golden stripes on the body and on the anal and tail fins, as you have seen in this pretty picture. Pigment cells in zebrafish, melanophores, iridophores, and xanthophores, originate from neural crest-derived stem cells associated with the dorsal root ganglia of the peripheral nervous system. We discovered that the stem cells are pluripotent and give rise not only to the 3 pigment cell types, but also to neurons innervating the skin and glia. All 3 cell types interact in a complex manner to form the alternating dark and light stripes in self-organization. The color patterns in closely related Danio species are amazingly different, ranging from vertical bars or spots or combinations of spots and stripes to no pattern at all. Their variation offers a great opportunity to investigate the basic genetic basis of color pattern evolution in vertebrates. Exciting technical developments of the recent years, especially next-generation sequencing technologies, and the novel possibility of genome editing with CRISPR-Cas9 system allow to expand from model organisms into other species and directly test the function of genes by targeted knockouts and allele replacements. Thus, models and hypotheses about pigment pattern formation derived from zebrafish can now be tested in other Danio species. These studies will lay the foundation to understand not only the genetic basis of color pattern variation between Danio species, but also the evolution of color pattern in other vertebrates. In my talk tomorrow, I will highlight our recent exciting findings. For those who want to know more details about this fascinating topic, I have published a small book called Animal Beauty: Evolution of Biological Aesthetics at MIT Press. Thank you very much for your attention.
Hi Yanni, many congratulations on being awarded your honoris causa in Barcelona. I hope you have a fantastic day, and I very much look forward to seeing you again once this whole pandemic is over. Bye.
Yanni, congratulations on this wonderful award. I just wish you could be there in person. And thank you again for all the wonderful science you've done and wonderful times in Tübingen. Have a wonderful day. Congratulations.
Dear Yanni, congratulations on your award. Yet another one. It was a privilege working with you, learning from you. Those memories I cherish and the lessons that I learned motivate me to be a good scientist all the time. Thank you.
Hi, Yanni. Many congratulations. This is a well-deserved honor, and I hope you're enjoying the ceremony. All the best, and congratulations, and my admiration, as always.
Liebe Yanni, herzlichen Glückwunsch. Dear Yanni, congratulations from me and from the whole lab. Liebe Yanni, herzlichen Glückwunsch. Herzlichen Glückwunsch. Congratulazioni.
Herzlichen Glückwunsch, Yanni. Congratulazioni. Herzlichen Glückwunsch.
Felicitaciones. Herzlichen Glückwunsch, Yanni.
Doctora Nüsslein-Volhard, doctora honoris causa de la UPF, senyora Montserrat Vendrell, presidenta del Consell Social, anterior rector Jaume Casals, professora Laia de Nadal, vicerectora de Transferència de Coneixement, professora Cristina Pujades, professorat, membres de la comunitat universitària, personal de gestió, amigues, amics. Sabem que la doctora Christiane Nüsslein-Volhard té diverses passions i una d'elles és la lead music, la música lead, i per això hem tingut aquestes cançons. We know that you have several passions and one of them is the lead music. That's why we have had these several songs. So we appreciate a lot the singers, the artists and the core, UPF core, for these marvelous songs. Professor Pujades Laudatio has stressed on many aspects of the Dr. Christiane Nüsslein-Volhard. For example, the scientific accomplishments, her role as a scientific mentor, and her commitment to closing the gender gap. I will focus my speech in this last part. You know that UPF is committed to several key pillars. One of them is that this is an intensive research university. Another is the internationalization. Another is the commitment to culture and humanities. And another is the commitment to planetary well-being. Planetary well-being means the well-being of the planet, of course, but also the well-being of organizations, the well-being of people. And closing the gender gap is related to this last commitment. And as Professor Pujades has mentioned, one of the accomplishments of Dr. Christiane Nüsslein-Volhard is the development of a foundation, the Christiane Nüsslein-Volhard Foundation, to encourage young female scientists to pursue and to have success in their scientific career. So this means that the honorary doctor we have, the new honorary doctor we have at UPF is a scientist, but also a person that, apart from being a great scientist, a person who has devoted her time to improve the planet, not only with her research, but also with actions like this foundation. UPF has, in biomedical science, we have a proportion of 50-50 female-male students. In medicine and biomedicine, but when we analyze the proportion, when we talk about the researchers that enter in this university, the 50-50 proportion changes, and now we have 25 female percentage and 75 percentage for male. So for students, the average is okay, but not for research positions. But in the whole university, the proportion of male and female for students, it's 80% female. I am talking about undergraduate students, 80% female, 20% male. Probably some of you, many of you know the reason for that, but some of you maybe don't know the reason. Why, for undergrads, in the general, in all the courses we have at UPF, why we have 80% female, 20% male? And the reason is that to enter in this university, we use the general system organized by the Catalan government, where the grades of the previous studies are crucial. So the students with better grades have more possibilities to enter in this university, which is probably the university where it's most difficult to enter. And you know that when female and male have 18 years old, 17 years old, 18 years old, male are thinking other things, football, soccer, and many other, and female are more concentrated in the studies. So probably this is one of the reasons. But what is very shocking is that when we analyze what has happened with this percentage along the years, and several times UPF has studied the evolution of this 80% female we have in undergrad and 20% of male, we have studied what happens along the years. And it's shocking to see that in the first year of undergrad studies, the performance of female is very high. In the first year, the performance of male is lower. But when we arrive to third year, fourth year, the performance is more similar. When undergrads finish the studies and they are graduated and they start working, the difference in performance starts to change. So at the beginning, a female performance is very high, performance of male is very low. During the career, the performance of female starts to low down in the third year, fourth year, and the performance of male is going up. And in one of the studies I remember, prepared by the Social Council of the university, the two key factors of making the performance of female go down and the performance of male go up is the different proportion of work at home. And how couples and families distribute their time. So this means a big problem, because many studies demonstrate that an organization and a society is better when there is a gender balance in top levels of organization. Many studies demonstrate that companies, even listed companies in the stock exchange, they have higher profitability when they have a gender balance on top. Higher profitability, higher performance, so many good results. So that's why we think that it's very good what you've done with your foundation to try to improve the gender balance. Here at UPF, we try to do our best to that, but it's very difficult to improve. We have slight improvements, but if we compare our first gender balance plan, we started more than 10 years ago. At the end of the first gender equality plan, we had some slight improvements. We have a second gender plan. Now we are about to finish that plan, and now we are studying the results, and the results are not good enough. So it seems that there is a lot of room to do in order to improve this. And as I have mentioned, we are devoted to planetary well-being. And planetary well-being means not only solving economic issues, the issues of environment, but also this big issue. Because remember, we have 80% of students in the undergraduate students, but when we see 15 years later, what are they doing? They are not in top positions. So this means that it's more difficult to make a difference in connection with organizations and so. It's true that everybody can make a difference whatever they are, but this is a big problem. That's why I congratulate you, not only for your research, but also for devoting some time to help young women researchers to pursue with their career. Here, for example, I have data. If we talk about Nobel Prize, from 1901 to 2021, only 58 women have been awarded, 58 out, and 889 men. This means 6%. At UPF, you are the 18th scientist to be, or person to be awarded with the honorary degree of this university. You are the 18th. Out of 18th, the first one was Desmond Tutu. And the last one is Angela Davis. And out of 18, we have 5 honorary doctors. You are the number 5. So this means more or less 25%. But there is a lot of work to do also in this field. So I wish that in the coming years we can have more improvements in all the issues related to the gender balance. I'm about to finish. So it's a great honor for us to have you as a new honorary doctor. It's a real great honor, and that's why we are so happy today. And thank you very much to all of you for attending this event and also to be with us today. Thank you very much. Ara ens hem d'aixecar, em sembla. Sí.
Gaudeamus igitur juvenes sumus. Gaudeamus igitur juvenes sumus. Post jucundam juventutem, post molestam senectutem, nos habebitus, nos habebitus. Ubi sunt qui ante nos in mundo fuere, ubi sunt qui ante nos in mundo fuere, adeas ad inferos, transeas ad superos, osi videre, osi videre. Vivat academia, vivan professores, vivat academia, vivan professores, viva membrum quod libet, viva membra quae libet, sempre sit in flore, sempre sit in flore.
Sentem un moment. Seiem un moment. Aquí. Estem a punt d'acabar l'acte. Ara esteu tots i totes convidats a l'aperitiu que servirà a la plaça interior del Parc de Recerca Biomèdica. S'aixeca la sessió. We have come to the end. Thank you very much for your attendance. We invite you, all of you, to have a drink at the Pierre Bibi in a square. The ceremony is over.